Good evening all. Let's wait for students to join, then we'll start the class. Today is the third class in our course, in our free marathon series, right? Let's wait for students to join, then we'll start. Good evening everyone. Please just confirm whether the audio and the presentation is clear or not to each one of you. Just confirm please in the chat sections. Is everything well? Audio, video? Let's wait for just one more minute, then we'll start with our class. Okay, thank you Harshit. Others also, please just confirm from your end. Okay, thank you Shubham. So we have 6 students in the class. Very good. Let's begin with our class. So today is the third class in our course, right? In our second marathon, which is running free on Botany Insider. And today we'll be solving out few of the previous year questions. First, I'll give you a quick revision of what we have discussed yesterday, and then we'll solve out the question. Before that, we are starting off with a new course, which is going to be launched very soon. If you are targeting out the June 2023 examination, this course would be a full package for you. We'll be solving out questions, we'll be practicing general aptitude, and all the theory units would be discussed in this course. It would be a 3 months course. So if you wish to enroll, we are about to launch it in a day or two. Just wait for the official notification. Okay. Then if you're new to Botany Insider, do not forget that we have our own application. You can download our app from the link given below. It is available both on the Apple Store and the Google Play. And these are the resources available to you all. We have theory classes, we have general aptitude course, we have notes booklet, PYQ pointers, practice session which is going on at this particular time. This is a live course which is going on. You can have the recording of other things as well, and the presentations are also available to you. Now let's discuss about the 3-point cross problems. Okay, before that, let's just quickly revise whatever we have discussed yesterday, right? I'll just give you a question so that you can recall every single thing, and then we will be solving a question. Now yesterday we discussed about 3-point cross, right? And we discussed about that there are 3 main outcomes when we look into the 3-point cross. What does 3-point actually means? It means that there occurs a cross between 3 genes. So I'll give you a question because we have discussed all the things yesterday. I'll give you a quick question. Write down all the possible outcomes, resultants in the chat sections. So just wait a second. Yeah. So let's say it is capital A, capital B and capital C, and over here we have small a, small b and small c. So write down what would be the resultant if a cross occurs over this particular point, that is between this point. What would be the resultant in the first scenario? Get the answer quickly in the chat sections, everyone. Quickly write down the resultants in the first case. If you have watched yesterday's class, it would become very easy for you. So write down quickly. Then we'll be solving out a question. Quickly write the answers. Capital A, small b, small c. Very good. Small a, capital B. Sadaf, check the second resultant. Harshit, please check the second resultant. First resultant is correct. It is capital A, small b, small c. Check the second resultant, please. And others also, please participate in the class, because today we have to do the complete calculation thing. So it completely depends upon how much interactive you are in the class. So make sure that you are writing down the answers. So for the first resultant, it would be capital A, small b, small c, and small a, capital B and capital C. So Sadaf and Harshit, it is in the second resultant, it would be capital C, that is this one. Okay, yes, very good. So this is what? This is our single crossover one, right? For the second one, let's consider that there is a cross occurring at this particular point. So neglect this. Now a cross is occurring at this point. So what will be the answer for this one? What would be the resultant over here? Quickly write the answers. For the second scenario, write down the answers now. Just a second. Just a second. It got vanished. Capital A, capital B, capital C. First is write down for the second one, everyone. Second resultant, if it is happening over here, get the answer now. Okay, yes, it would be capital A, capital B, small c, and small a, small b, and capital C, right? So this would be the second resultant. This is what? This is our single crossover 1. This is our single crossover 2. In the third case, what will happen if the cross occur between both of them simultaneously? Now what will be the answer? Quickly write down, and everyone please participate. Those of you who are in the class, please write down the answers. This one would be double crossover. So what will be the answer for this one? Quickly write down the answers. Yes, what is the answer for the double crossover concept? Over here, what will be the answer? Please participate in the class. Yes, it would be small a, capital B, small c, and capital A, small b, and capital C. Very good. So this would be the resultant. So this is what we have discussed yesterday about the 3-point cross. Let's quickly discuss one question, because I don't think much of the students are interested in solving out the question. We can discuss one question, that would be enough for all. Okay, so this is a previous year question, right? This was asked in one of the paper in CSIR. The question was that you were given all of these things, and then you have to find out the map distance between capital A, capital B, and capital A, capital B, and capital C. So we have A, B, C, 3 of the genes present, and it was asked to find out the distance between A, B, and then to find out the distance between B and C. Okay, so let's try to understand how to do these type of questions. Now, first thing first, that these are the number of progenies. Okay, remember one thing, that the maximum frequency, that is 300 and 310 according to this one, this is what the maximum frequency will always be of the parents. Okay, remember this thing, this is a thumb rule which you need to remember. So what are the parents in this particular scenario? We have capital A, small a, capital B, small b, capital C, and small c, and this is the parent 1, and the second parent is small a, small b, small a, small a, small b, small b, small c, and small c. So this is the second parent. So these two are our parents. Now when you have to find out the distance between capital A and capital B, what you have to understand is, let me put a okay. So first thing first, we have capital A, small a, capital B, small b, capital C, and small c. This is parent 1, and this is small a, small b, small b, small c. This is parent number 2. What are their frequencies? It is 300 and 310. So just write down 300 and 310. Okay, so these are our parents. What will be the genotype of them? It would be capital A, because capital A would be expressed, then we would be having capital B, because over here it would be expressed, and then we would be having capital C. In this case, we would be having small a, small b, and small c. Now talking about that, what would be the resultants? We have just discussed the same thing. Same thing would be happening over here as well. So we have small a, small b, and small c. So single crossover 1, when you need to find out the distance between the first two genes, because over here it is asked to find out distance between capital A, capital B, and then capital B and capital C. So when you have to find out the distance between the first and the second gene, what you have to remember, that you have to take the single crossover 1 frequency. So resultant of single crossover 1 would be what? It would be capital A, small b, and small c, and second one would be small a, capital B, and capital C. In the second case, it would be, for the single crossover 2, it would be capital A, capital B, small c, small a, small b, and capital C. And for the double crossover, we would be having capital A, small b, capital C, small a, capital B, and small c. Right? Now what you have to do, always remember then when that when you have to find out the map distance, this is the unit, it is mu. When you have to find out the distance between the first two genes, what you have to take? You have to take the frequency of, or the number of the progenies of the first crossover, because it is between the first two, then the double crossover, divided by the total progeny, okay, or the total frequency multiplied by 100. And if that is for the percentage, but for the map distance you have to do it in this manner. Then if you need to find out the distance between second and third, that is B and C, what you have to do, same way, map distance would be calculated single crossover 2, that is the number of progenies in the single crossover 2, which is the second one, plus the double crossover, it would be added in both, divided by the total. So then you will be getting the map distance between the second and the third one. Now let's just analyze over here according to whatever we have discussed. So this one and this one is what? These two are our pair, sorry, this one and this one. This one is and this one, both of these are our parents. Now analyze among the others the single crossover resultants. Which one would be the single crossover according to what is given in your screen? Analyze and get which one would be the single crossover. The resultant that we have discussed just now, it would be what? Parent is ABC, small a, small b, small c. So it would be capital A, small b and small c. Second would be small a, capital B and capital C. Analyze what is the frequency over here. Observe very carefully and write down the frequency of the single crossover in the chat sections. Yes, analyze and get what will be the answer over here. What will be the frequency? 16 and 14. Very good. So single crossover is 16 and 14. In addition to that, analyze what would be the double crossover as well. Analyze what will be double crossover. Sadav, analyze properly. Capital A, small b, small c. Capital A, small b, small c. So it would be 120. Observe carefully. Then it is small a, capital B, capital C. So small a, capital B, capital C. So it would be 100 plus 120. It is not 16 and 14. Analyze properly. Okay, let's do one thing. I'll be doing the complete question because none of you is ready to answer it. Absolutely fine. Let me do the complete calculation because we just cannot wait and for this thing. Okay, so for single crossover it is capital A, small b, small c. So it is capital A, small b, small c is 120 and then small a, capital B, capital C is 100. So for the single crossover one it is 100 and 120. For the single crossover 2, double crossover is 16 plus 14. Yes, very good. And for the last one it is 65 plus 75. Okay, so these are the three frequencies that you need to remember. Now when you put it into the formula, this is how we will be actually getting it. So for finding out the distance between the single first gene and the second gene, it is double crossover plus single crossover upon the total crossovers, that is the total frequency into 100. So it is 16 plus 14 plus 120 plus 100 divided by 1000, because when you count all of the given one, it is equal to 1000 and multiply by 100. So when you calculate, you will get the answer as 25 mu. Then second one is distance between B and C. So for the second and the third gene, it would be 16 plus 14, that is a double crossover, right? And this is the single crossover 2 divided by 1000 into 100. So it would be 17 mu. So according to the question given to us, answer would be option number A. That is option number A will be the correct answer for this particular question. I hope this question is clear to everyone, because no one is interested in solving out the question along. So I have explained the question myself. I hope this is clear. If you have any doubt, any problem, please ask. Okay, so I guess this particular question is clear to everyone. I had to discuss many more questions as well, but students are not participating in the class and we cannot just do maths in this manner. Okay, so this was about for today's class, I hope. Sadaf, that is a different question. For this particular question, it is only for the map distance. It was only for the distance between AB and BC. Okay, for gene order it is again very simple. Like, see, over here you are given the frequency. In the gene order concept, what it is given, you are given the frequency, but you are not asked about the distance. You are mainly asked about that what will be the actual gene order. For that there is a different complete concept. We will be discussing it in some other class. I don't think students are much interested. I have bought so many questions, but let's do it tomorrow. Okay, so this was about for today's class. Thank you for joining in, everyone. Thank you for your precious time. Let's meet tomorrow. Okay, good luck. Take care, everyone. This was the question that we had to discuss. There were so many other questions to be discussed, but none of the students are participating and we cannot proceed in this manner. Let's meet tomorrow. Bye, everyone.